will just be a brief talk about cell types and the way that we define different kinds of cells here at the Allen Institute, and also a brief history about how cell types have been defined over time, since this has been an ever-evolving problem for like the last 130 or so, 40 plus years in neuroscience. So how can we define cell types? Well, one way to define them is that they're groups of cells with more similar quantitative cellular properties to each other than to other cells. That's sort of a vague overall way of describing that. And they tend to also share similar features defined in usually more than one or more measurable criteria. And again, those are both pretty vague answers because we currently lack standard definitions of cell types in the brain, right? We heard some people earlier talking about their favorite cell types. Some people said some commonly said things, commonly known names. Some people might have used different names for the same type of cell, right? That's just an ongoing thing that we have here in the neuroscience field. And so this is through, a product of evolution, the genetics and the development of the environment that form these cell types. And then how do we define these cell types, right? We have anatomy, morphology, electrophysiology, connectivity, and overall, what's their ultimate, spiking activity and function. So there's a lot of different pieces to this puzzle. And historically, right, we have defined cell types in neuroscience by their morphology. We've been doing this since the early 1900s, the late 1800s with Ramon y Cajal. We still use some of those names today. We look at pyramidal cells, we look at chandelier cells. People outside of neuroscience might be thrown by the name chandelier, but we just say, hey, it literally looks like a chandelier. And that's why we call it that. But now we know more information about those chandelier cells. But right, we still use these morphological names. We still say basket cell. We still say bushy cell. Martinati isn't named actually after, it's named after a shape, but the Martinati is not a shape. That's just the last name of the guy who discovered it. But that's a type of somatostatin cell that happens to have a particular pattern with its axons to layer 1. And again, we still use these names, right? These are names that we've been using for many years. But now there's more information that we can find out about these particular cell types, right? We can also figure out how they're connected to each other. We now know that those chandelier cells, right, they have a different connectivity pattern compared to other types of neurons. They target those axon initial segments. They're very potent in their inhibition. Meanwhile, our basket cells, yes, they look like baskets and they sort of envelop other cells, but they also target cell bodies. That's sort of another lens through which we can look at these cells, their shapes and also how they connect. And then that's just really looking at our inhibitory cells, but we also have names based on all of our long-range projecting cells, right? Our excitatory cells, our intertalencephalic, our extratalencephalic, our pyramidal tract, our corticothalamic cells. Again, this is all cortex-based because that's kind of where a lot of the cell typing has started, was really in the cortex. So all of these really refer to just different areas in different parts of the cortex that they're in and different other parts of the brain that they project to. So intratalencephalic, they stay within the telencephalon. Extratalencephalic, they go outside. Corticothalamic, they go project from the cortex down to the thalamus. And we also have thalamocortical cells, right, that do the opposite. They start in the thalamus and they project up to the cortex. Electrophysiology. We have a couple of electrophysiologists in the room. They're going to have perhaps completely different names how they refer to their cell types. They might say, oh, it's a fast spiking cell, or that's a intrinsically bursting cell, or that's an adapting cell, which might not make sense to people who do not study electrophysiology, right? But over time now, we've been able to link those particular names based on those firing patterns to specific morphologies or to specific transcriptomic types. And that's still a really big area that's very active in neuroscience, is really linking that transcriptomic type to the electrical function here. And historically, we've also used a lot of molecular markers. So especially in the cortical inhibitory neurons, it's been known for a couple decades now that there are these particular proteins that happen to be expressed in these different groups and not in other groups. They tend to be non-overlapping between, the parvalbumin, the somatostatin, the vasoactive intestinal peptide groups. And then there's a bunch of other ones that are sort of intermixed. We learned about CCK earlier. Someone mentioned CCK. We have NPY, neuropeptide Y. We have calretinin, CR. So basically, we just have all these different molecular markers. And when we look at our excitatory cells, we don't a little less is known about which particular markers are in there. They're more known by their projection types. And again, this paper is from the 90s. So, the big issue is connecting all this historical data, right, into something together and concrete that makes sense today in 2026. How do we combine all these different types of data and integrate it together? And so people have tried to do that. Here's a chart from a review paper from 15 years ago now, trying to just sort out how do we connect these molecular types with the electrophysiological types and their morphology. Do these things make sense? Do they connect? Is one ephys type consistent with a molecular marker? Sometimes yes, sometimes no. This chart was updated, a couple of years later by that same lab to expand upon that. And then it was updated again, a couple of years ago where they actually said, oh, there's actually another molecular marker for this other group. And, so this is just to show this is an ever expanding issue in neuroscience. Another example here is another review paper. Everything I just showed in that past slide was looking at cortical inhibitory neurons. Here we're looking at our cortical excitatory, specifically our extratelencephalic and our intratelencephalic. I mentioned earlier that they tend to be named by those projection patterns, but now not only do we have those projection patterns, we're starting to get molecular markers for these guys as well, which came many, many years after we had those molecular markers for all the previous inhibitory cells. And why does all of this matter? Why are we studying cell types? I think this group knows that, there are many different functions and modalities for different cell types, right? Just because they both express the same marker or maybe have the same firing pattern doesn't necessarily mean they have the same function, right? You also take into account where they're located, where they project to, what other types of cells, synapse on that particular cell, right? How does it fit into the circuit? It's much more complicated than just naming cells. The next step is also figuring out what are they doing, how do they connect to each other, and also then how do they change with disease, right? And if there are certain groups or subgroups that happen to be affected in disease, why is that happening? And so all of this is just to throw out, all the different types of techniques and work that has been done before single cell transcriptomics, right? Everything that I mentioned previously was all different ways that neuroscientists have been really studying these cell types from different modalities without being able to really get that single cell data, right? Using antibodies, using, transgenic mouse lines, retrograde tracing of projections, the morphologies. But then now in the last, 10, 15 years or so, with the advancement in new technologies, specifically with single cell RNA sequencing, now we're able to get much more information, a lot more richer information about each cell types. But then now we have to connect that new information back to the previous historical information, right? Because that information is still extremely important. And now you have to try to match it up with the current data. And so that's something that we're really working on. And just to show again that there have been many conferences over the last couple of decades trying to figure out how do we even define cell types. This was a group that met back in 2008 in Italy, I believe, coming together of saying, how do we just come up with a naming schema for these different cell types? How do we do it? And this was a large list of people that they invited. And they wrote this paper at the end, and we wrote, we hope that this is the first step that will later lead to a broadly adopted classification. However, before we are able to speak a common language, we need to agree on the words. And this is from 2008, and this is still, still an issue in 2026. And so how are we working towards this issue here at Allen Institute? Well, we're primarily relying on information from that single cell transcriptomic data to really determine the cell types, and using that as sort of the anchor, and then taking that other information based on the projection data, the morphology, the electrophysiology data, and sort of adding that on to the transcriptomic data, but really using that transcriptomic definition as the anchor. That is the model that cell typing is done here. And why transcriptomic data? Well, it's very high throughput, especially with the newer technologies. You can measure, many features of the cells, so over 30,000 genes. It's very helpful for working with postmortem tissue, right? So a lot of the work we do here at the Institute is some human postmortem tissue. And so that's very helpful, right? And also mouse tissue as well. It works across different organs and species. Again, I just mentioned we do this work on different species, and there's a lot of different analysis tools that exist out there to analyze these data. And here's just a graphic explaining, again, mapping those different types of data, whether that's electron microscopy data or looking at synaptic physiology or anatomy data, and really anchoring that on the transcriptomic data to define our cell types. But another issue is that we still need a standard taxonomy format, right? Once we have all this information and we're getting it all together, how do we formulate it in a standard format that other researchers can use? So back in 2019, there were many different representatives of neuroscience who came together at the Allen Institute to have a group meeting about this. So similar to that 2008 meeting back in Italy, there was one here in Seattle showing that something that we're still, working on, except for here where this was looking at really naming, coming up with a concrete naming system for different cell types and coming up with a format for that taxonomy. And this was the result. The results are also published in Miller 2020. Jeremy's also sitting there in the back, so I'm sure you can also ask questions to him about this afterwards. But yeah, the result was the Common Cell Type Nomenclature, or the CCN. And so what's shown here is sort of an overall example of what that looks like. And first, the schema and also the naming convention. And this is specifically starting in the mammalian cortex. So the idea is you have this taxonomy, right? We have this dendrogram here, we have all these different branches, and we have all these different cell types. And in this dendrogram here, this is based on transcriptomic data, and the cell types that are similar together in this tree are more similar transcriptomically, and those that are farther away, right, are much more distant. And we have all these different taxonomies for different kinds of species and also different brain areas. And so the taxonomies themselves, the collection of all the cells and how they fit together, that hierarchy, also must have a unique ID, aliases, and other metadata to allow tracking of information between different studies. So why is that important? I'll show you in our next chart that we have many taxonomies that sort of build on top of each other. So you need to remain consistent with the naming between different versions. And then actually naming the cells in the actual taxonomies themselves. So I mentioned previously that, our GABAergic, our inhibitory cells already pretty much have non-overlapping genes that have been known throughout, from the literature, and those are still very much used in the taxonomies. We're not going to rename parvalbumin cells because people know what they are and they're pretty well known. And the glutamatergic cells, right? Those excitatory projecting cells, that's still maintained as a format here, saying which layer they're in, the projection, and then our non-neuronal cells as well, and any other historical cells here, right? Like here, we're not throwing out chandelier cell. It's still in there because it is a well-known cell type. And so what do I mean about all these different taxonomies? So here's an example of almost, I believe, all the taxonomies that have been produced through the Allen Institute over the last decade. So this starts with back in 2016, there was one in the mouse visual cortex, which had, I believe, like 1,600 cells in it using SmartSeq sequencing. And then our latest whole mouse brain taxonomy has total 8 million cells in it between single cell and spatial transcriptomic data and over 5,000 different cell types. So this is just sort of showing the evolution of all these different projects that have been going on at Allen Institute. And there are a lot in mouse, sort of the model organism, and then we've scaled up from mouse to incorporate non-human primates, so marmoset and macaque, and also human tissue as well. But mouse has really been the anchor of that. And so the way that we've been able to work with the other species is by doing these consensus taxonomies. So in 2021, there were a number of multi-species, cross-species taxonomies that were produced. And should also mention, this isn't just Allen Institute. This is, I mentioned before, BICAN, the Brain Initiative Cell Atlas Network. Prior to that, it was the BICCN, which was the Brain Initiative Cell Census Network. So that's just been an ongoing consortium that the Allen Institute has been part of that's funded through NIH. So these are all of these come from that consortium work. But yeah, over the years, we've been focusing on first smaller brain areas, right, in individual species. And then, there was a human study in middle temporal gyrus in 2019 from Rebecca Hodge. And then, there were other studies then comparing that, right, with other species. And then once you have one area, we started with cortex, then how do we get to subcortical areas? Then how do we expand to the whole brain? And then once we have the whole brain, how do we expand that out to either an aged brain or a developmental brain, an embryonic brain, or, a diseased brain, right? How do we branch off from that standard that we have from our healthy young adult brain? And so there are many papers associated with all of these different taxonomies. The link to this might be already in the GitHub repository that I mentioned. If not, I will add that after this talk, but just linking to all these different papers. So these papers are really the focus of like the next 3 days. These are all the studies that have these different taxonomies that you can either map your own transcriptomic data to, or you can visualize them, or you can access them programmatically and do your own analysis on these studies. But especially just in the last year, we've really branched out for creating an aging mouse study that was published last year, Kelly Jin 2025. We have a basal ganglia consensus taxonomy for human, marmoset, and macaque that was just put on bioRxiv. And some of the speakers, the lead of that study, Nelson, will actually be here this afternoon to talk about his work. And then Matthew Schmitz is the first author on a mouse, macaque, and human consensus spinal cord taxonomy, which was also just put online on bioRxiv about a month or two ago. So these are really just the anchor and the backbone, just summarizing all of the different cell types that we have here. And what does a cell type mean in each species and each brain area? And then are those cell types conserved or not in all these different areas and all these different species? Sometimes yes, sometimes no. So how do we visualize all these big taxonomies? So you've already seen a dendrogram in some of the previous plots, right? Depending on the size of the taxonomy, sometimes you can actually show all the individual cell types. So in this one at the top, this was mouse primary motor cortex. There were only 100 or so cell types identified. So we could actually show all 100 of them in this dendrogram tree. But down here at the whole mouse brain, right, there's over 5,000 types. So we can't show all of them in the full paper. This is only going down to the 300 level, but if we keep branching, you get all the way down to the 5,000 level. And so here are those same different taxonomies. Again, we have the motor cortex taxonomy that I just mentioned. It was a dendrogram. It was that top figure in the previous slide. Now on the left, it's the sunburst plot where we can see all those different kinds. In the outer ring, you see 116 different types. Here in the whole mouse brain, now we have a UMAP, or uniform manifold approximation and projection, which is showing us, it's colored now to show us the 5,000 different cell type clusters that were identified within the 4 million different cells. So these are just a few different ways to visualize extremely high dimensional data, right? Because how else do you show 4 million cells that have, 30,000 different genes? That's a difficult task in itself. And what to do with all these different taxonomies? What's the point? Well, you can use it for your own data. You can use it for data annotation. I mentioned previously, if you're doing your own transcriptomic work and you say, okay, I found cell type parvalbumin 12. I want to know what the Allen Institute name for that cell type is, right? Here's just an example from something I found in the literature where we actually have a tool called MapMyCells where you can actually use that tool to annotate your own data. And again, we'll go over one of the engineers who made the algorithms in MapMyCells. He will be here on Wednesday. So if you have specific questions on that, we will have a lot of time to talk to Scott on Wednesday about that. But also, not everyone does transcriptomic work, right? is a paper from my PhD work before I worked at Allen Institute. I was not doing single cell transcriptomics. I had a lot of mouse lines and a lot of antibodies. And the goal was, okay, these are the tools I do have. How can I use this transcriptomic data to inform my decisions and how I define cell types with, the tools that I have. And so I use that transcriptomic data to pick, 6 different mouse lines that I was working with that labeled different cell types, and then intersected them to figure out, different groups of cell types based on that transcriptomic data. So even if you don't work with transcriptomic data yourself, there are many ways that these data sets can still be useful. Here's another example of that. I think I showed this paper earlier, but this was a group that, they just scanned, mined all of that literature and data and said, okay, based on all this information from Allen Institute, seems like we have these kinds of groups of neurons, but there's really not a marker for this other group. And by just looking through all this data, we just found another marker for this group and we're going to publish about it, right? So just because we're publishing all these large data sets doesn't mean that people at Allen are going to find every single bit of information in all those data sets, right? That's for everyone else in the world to work on, to go through those data sets, find those interesting things, study those specific genes or cells or, brain regions and dive deep into those, right? There's tons of areas for exploration. All right, and that is the end of this talk. We can take some questions. We have a couple minutes for questions, and then we're also going to move on to a fun activity before our next talk. So do we have any questions about anything? Yes.
I get into this later, but I was wondering what's the exact terms of this publishing based on this?
Yeah, we have a citation policy. I can show you where that is on our website. But yeah, basically, if you just you can cite the tools because some of the tools have their own RRID. You can cite the papers, and some of the individual data sets also have their own ways to cite. That's a question we do get a lot, is how exactly do I use it and cite it? I mean, yeah, it's open use. And like, yeah. And some people don't even cite things correctly, and then I just search through on PubMed and I misspell or I type in the wrong, because sometimes people, accidentally miscite things. So I try my best to find all the things anyways. Yep.
Some kind of collaboration with English.
If it's we have a whole legally licensing team and you can shoot them a message and they'll figure out those bounds.
Yes, we do. I can show you the link to that. Yeah, because that is a question. Because it's, yeah, the licensing for fair use, as long as it's nonprofit work, which is, most academic work. So.
One of the questions that you had brought up in the intro as well was about whether cell states or cell types are transient or stable. And I know with transcriptomic data, when you draw out the clusters, you kind of infinitely make the clusters more detailed and smaller. How do you balance like separating out a cell type, defining that versus like looking at it in pathology versus normal states and how those might interact?
I will say I'm not the best person to answer this, but we will have 2 people this afternoon. If you bring up that question again, or I'll bring it up, someone will bring it up. Yes, those who are deep in the weeds with doing the actual analysis and defining the clusters, they will have a good answer for you on that.
Don't know if it's still available.
Yeah, SFN like deleted them after something.
Ongoing question, the field of cell space. What do you think of the 2-sentence summary of that discussion?
I think the 2-sentence summary is that a lot of people are really interested in understanding the difference. The computationally measure the vote basically the same way, so it's a big semantics the way that we do most of the time. So I guess that's my it's important biologically, but hard to distinguish. It's not impossible to distinguish.
Yeah, and I think, yeah, for this afternoon's talks with Nelson and Morgan, that will also be a useful thing.
A lot of people. Sometimes.
That's true. It's one of those like who you ask type things. Yep, another question?
Outside, so let's say about data was something in the actually in that panel, it doesn't look exactly what we see, or we see like region commonalities that we just go away and go ahead and publish it, or should we look for the communication first and I don't know if that makes sense.
No, I think like from what I've seen, I mean, yeah, I've definitely seen, yeah, papers that are maybe critical or in disagreement, right? You know, I think that's just part of working in science. Because the groups that we do work directly with and publish our work with are defined through our collaborations and like the consortiums that the Institute is part of. But another option, if you're just curious about like dialogue and, you know, conversation, we do have a community forum. It's, I'll type it up and I'll link it to the GitHub repo, but it's community.brain-map.org, or you can just search like Allen Institute Community Forum. And we're on there all the time, like just answering people's questions. Oh, I just kicked something, sorry. But if you post something in there and you're like, oh, I found this thing and I, you know, maybe this is this, someone at the Institute might, you know, that might open up a door for some conversations. Jeremy's on the forum a lot, answering things.
I'm sorry.
If someone external is doing something and they find something that is maybe not quite in agreement with like an Allen Institute taxonomy, they find something new, they find something different, and they just, he's like, oh, should we publish that first or try to talk to Allen first? I'm like, well, publish it. Like, yeah, we're not stopping anyone from that.
I think that will both allow people to link to be and then better talk to us happier is fine. Letting us know, you know, that can encourage. We will at some point do cell type definitely data, and we'll definitely.
All right, yeah, another question.
Just like a repo web, say you generated a new single cell RNA-seq data set in some specific brain region. How would you go to this point to finding cell types? Like what's the pipeline for it?
Yes, well, actually, we're gonna have 3 different talks this afternoon that sort of go through in detail, like more of each step of that. So I'll let them do it. Yeah, this was just more like an introduction, sort of a setup of like the rest of the talk. So like we're gonna have people come in talking about many of those different taxonomy projects that I mentioned throughout the next couple of days. And yeah, this afternoon, we're actually hearing about our RNA sequencing pipeline. We're also going to be hearing about how to do the cell clustering and build those taxonomies, and also how to do it cross species. So those are the 3 talks from this afternoon, and talking about the Brain Initiative and the Cell Atlas Network and our collaborations.
But today, instead, we're going to be chatting about something I think is a lot more fun, more near and dear to my heart, which is looking at all of these taxonomies now in an evolutionary context. And so, of course, as humans, we have these incredible complex motor abilities, the ability to speak, learn language, and the ability to use tools, both of which are fairly unique in the animal kingdom. And I think it's of interest to a lot of us, you know, how does this actually run in the brain? What's the sort of programming for it? And so, you know, just to step way back, a lot of the, you know, the groundwork to be able to approach questions like this started with the Nobel Prize for Ethology, which is just kind of the quantitative study of behavior. especially from these folks who looked at, Tinbergen's supernatural stimuli, Conrad Lorenz with imprinting, and Carl von Fritsch with the bees waggle dance that tells them, where the particularly sweet flowers are. And so that evolved then into neuroethology. And so neuroethology, which it sounds like some of you are already familiar with this, but it's the idea of studying the animal model that's an expert at the sort of behavior that you're interested in. And so, so again, if you're interested in language learning and speech, Songbird could be a really, attractive model for that, because they also have to learn their songs when they're little, in the same way a little kid learns language, and I've been able to find that there are a lot of shared genes and shared networks for that even our closest relatives, chimpanzees, don't share as vocal non-learners. But of course, there are other, a barn owl that can hear a mouse in, a completely pitch black barn and pounce on it, bat echolocation. There are all these exquisite behaviors in the animal world that we can learn from. And sadly, over time, especially with the advent of the genomic era, molecular genetics, we've sort of lost that thread a bit, where, inbred mice have so many resources around them. We were sort of limited by who we had genomes for. And I think there's also a bit of like, just inertia, it's easy, just study it in a mouse. But we lose a lot of that complexity. Thankfully, I think, more and more we have the opportunity to turn things around. We have, at this point, 22,000 animal genomes, and millions and hundreds of thousands of even really advanced epigenomic data sets that are showing you how the genome is becoming activated across different tissues and cell types, so that we can dive back into these animal models. We're working on a pretty cool project here, the Human and Mammalian Brain Atlas, which is trying to do exactly what Nelson said, not just to study these different species, for the fun of it, but because it's literally a useful tool for being able to confirm, oh, I see the same cell type across all these different species. So now I can actually be more confident that this is a real finding and not just some weird artifact of my pipeline. And so, yeah, so I would encourage you all to, to think ethologically, to think about, now that we're in the genomic era, what sorts of, more interesting models could I tackle, especially as you're making transitions from PhDs to postdocs. And, I think academia gives you a lot of opportunities to sort of explore these, I think, perhaps more exciting models. And I'd be happy to chat and give advice for anybody who's interested in this sort of thing. So for me in particular, I'm super interested in motor behavior, how the brain interfaces with this sensory world. And if you look at the brains of diverse species, you see that the brain structure actually reflects the sensory world that an animal lives in. A platypus's brain that has to, have this mechanosensory, view of, little worms wiggling under the ground is going to look different from a raccoon's brain that has this incredibly sensitive hand, for opening your trash can and, digging around. And so, as with anything in biology, we can study this at all kinds of different levels, down to the molecular genomic, to the transcriptomic, to the cellular, to the systems level. And so we're particularly interested in this project, the gene regulation, differential expression, and cell type abundance differences. So that's what I'm going to be talking about. And importantly, what's different when you're doing these species comparisons, these evolutionary comparisons, there are different kinds of comparisons we can do. And so on the right here, you can see on the one level, you can ask, what's conserved? In all the different animals I'm looking at, what do they all share in common? Do I see that same cell type appearing over and over again? Second, you can ask, what's diverging? do I see in rodents, suddenly one cell type becomes two? In primates, does a novel cell type emerge? We do see that. And third, there's convergence. So do two animals that have, vocal learning, like, again, the human and the songbird, where they've converged on a similar behavior. Or you could think of, a dolphin and a shark. You could think of countless examples of convergence in nature. Even, when you see this similar phenotype evolving, is the molecular structure, is the neural structure underneath also converging? Or are they solving the same problem with completely different pathways? And so this work started off with a 3-mammal taxonomy, and it's now expanding to a 25-mammal taxonomy, covering everything from, a range of primates all the way out to marsupials. And so this is the full list. Again, we're really interested in the sort of evolution of unique human traits, so primates are overrepresented, but we're also trying to get a broad swath of species from across the mammalian taxonomy. And this, of course, wouldn't be possible without a ton of collaborators that helped us to source some of these, precious rare tissues. And Nelson already talked to you a lot about the actual nuts and bolts of taxonomy building, so I'm not going to go into a huge amount of detail on that. But what I will say is that what was uniquely challenging about this cross-species work is that it's not enough to just build one taxonomy per species and do the donor correction. We also had to merge, repeatedly across all species, to try to clean up, okay, this thing that looks like a unique cell type, actually it's a dissection artifact, or, just some weird double, it took a long time to get all these cell types to play nicely in the right space. A big part of this is because, like Nelson talked about marker genes, right? Marker genes are a really powerful way that in any given species, we can identify a cell type of interest. Sometimes we even name cell types with their marker gene, right? The problem with that is marker genes are often not conserved. These are labels that we give to these cells, but the cells, they may not need that gene in any given species, right? Just because something is really uniquely active in one cell type doesn't mean it's necessary for that cell function, right? Marker genes, they're convenient for us, but it's not like a biological thing. A lot of times you can have one gene performing a given function in one species, but then in another species, it's like, oh, this cell type, this should be firing a little faster. So I'm going to swap out this potassium channel for a related one. And, and so the function is conserved, but your marker gene is gone, if that makes sense. And so we had to work around this and, to some degree, just be very cautious about being too adherent to any one collection of markers and rather try to take the whole picture of the transcriptomic fingerprint of a given cell type across species. So that's what took so long about that. But I'm happy to say, and this is one of those unexpected results that we were talking about. I thought that there would be a lot more like cell species specific or lineage specific innovations. And at the deeper cluster levels, there certainly seem to be. But at the subclass level, there's remarkable conservation across species of, pretty much all of these subclass level types. And so in the past, there's been questions about, for instance, this cool interneuron called a chandelier cell. It has this beautiful architecture where it's spreading out, in a big bushy way across a bunch of different brain regions to sort of integrate signals. And there's a question, is this conserved? Is this primate specific? People kind of bounce back and forth. And now we can definitively say, yes, chandelier cells are deeply conserved, both in terms of these, markers and in terms of the overall transcriptomic fingerprint. So that's pretty cool. And so one of the first things we were interested in is, okay, we have these conserved cell types. What kind of variations are there? And one of the primary axes they seem to vary along is abundance. You see with that spread there, side to side, there's different abundance of these cell types in different species. And interestingly, it seems to follow this sort of pattern of a shift between inhibitory and excitatory neurons. And so we're wondering, what could that be? And if you read the established literature, it's known that humans have this higher sort of EI ratio that, it gets explained as, having more inhibitory neurons, it allows our big primate brains to do all kinds of special computations that rodents can't do. It turns out that's a fiction. It turns out that actually it's rodents that are the weird ones compared to other mammals, where most mammals are more primate-like. But actually, if you follow the pattern even a little more carefully, it turns out that this is something that it's more of a scaling law. And so as you grow a brain, you need to change this ratio just to maintain the right EI balance between this sort of push-pull between excitation and inhibition. You don't want too much of the other. But just as you grow a brain, and here you can see the scaling, it actually follows quite elegantly. Just as a brain grows, you need to shift this ratio accordingly. And, and so these are just some more visualizations of it. But this is looking at some of these L5-ET projection neurons. So these are neurons that are, some of them are projecting across your entire body, to control distant parts. And, if you're a mouse, you just need a different density of those versus a large brain human. And so that's interesting, that scaling law in the glutamatergic, the excitatory neurons. But oddly, the interneuron abundances stay largely the same if you're comparing the presence of different interneuron types against each other. Except for finally some species-specific novelty in the opossum. They have a lot more fast-spiking parvalbumin neurons and fewer LAMP5 interneurons. So I don't know if we have any opossum experts in the room, but that's sort of a fun, open question that we'd like to figure out. And interestingly, getting back to the question of marker genes, again, there are interesting swaps that happen, where in human, this particular serotonin receptor subunit is a really good marker of a particular, kind of mid-layer, whereas in the mouse, it no longer is a marker of that type. It's like broadly distributed throughout the cortical column. So this is, again, just, a marker gene that you find in one species. No matter how beautiful it is, you can't assume that it's going to be a marker across the species. You have to actually look and find those conserved markers. And so when we put all the kind of median gene expression together, thankfully we do see the taxonomy pretty well replicated. Primates look like primates, rodents look like rodents, Laryngotherians grouped together too. So that was good to see. And now we can start comparing different DE genes across these groups. And so, this is sort of what this looks like. This is, this is still in flux. We're working on this as we speak. But one cool thing is sort of confirming and fleshing out an observation that other people have made, is that when you look at humans and, what are some of the gene specializations that seem to make us human compared to other species, it's not as much in the neurons as you might think it would be. It's actually the glia that overwhelmingly seem to be specializing. So oligodendrocytes, astrocytes seem to have a lot of human-specific specializations compared to any other cell type. So, this is something, it's been discovered in methylation data. It's been discovered in like human versus Neanderthal and Denisovan, SNP variants. So there are a lot of different pieces of evidence that are converging on this now. Okay, cool. So now we have these cell types, we have some differential genes. What else can we do with this? So Nelson talked a little bit about how you can integrate the spatial data, our collaboration with those teams. And so here we can take our L5-ET neurons, those, corticospinal projection neurons, and we can actually identify which is the cell type that's making the corticospinal connections versus like corticothalamo connections. And so, yeah, and so you can see this here with these retrograde labeled corticospinal neurons, where in the little colored image there, you can see the retro label signal overlapping the markers of that cell type. So that's really nice to see. We can also use some of these proximal enhancers nearby, some of these genes of interest to develop now synthetic enhancers so that we can specifically target species that are even quite divergent from human to mouse and all the way into ferret. And so these tools will also soon be available, which is pretty exciting. So the next phase, we're hoping to go to that third layer of convergence, where we have a good idea of what's conserved, what is starting to change in a lineage-specific way. But we're excited now to look into how these different interesting behaviors like vocal learning might be supported by some of these changes that we're seeing at the cell type specific level. And so this is a paper I had out that is exploring this to some degree, but this was just with the bulk attack data. And so it's going to be really exciting now to take these changes that we identified that are correlated with vocal learning on a genomic level and be able to ask with this new data set, what actual cell types are being affected here? I'm also really interested in, again, the evolution of dexterity. If we look at different primates, they have different levels of, thumb, forefinger precision grasp. Again, bringing back the dog versus raccoon differences in other species that are more or less dexterous. We see anatomical correlates of that, where the actual density of the projection of this corticospinal projection is denser in a more dexterous species. So the question now is, what are the genes that allow that dense projection to form? Do we see more axon guidance genes, netrins, semaphorins, things like that? What's the actual mechanism that supports it? So that'll be really exciting to get into. So just as a final take home, and then we can dive into as many questions as folks want to ask. If you take away one thing from this talk, it would just be, if you want to find out one of these evolutionary differences and you just have two species you're comparing, a lot of people sort of naively assume, oh, wow, I found a difference between humans and the mouse. So cool, I just found the human-specific, change that's making us so human-y. And that might be the case if you look across more species and you do in fact confirm that it's still unique. But equally plausible is that actually the state that you were looking at is rodent-specific. And because rodents, their intergenerational interval is so fast, it is more frequently the case that any difference between primates and rodents, it's more likely to be a rodent-specific thing, not a primate-specific thing. And, again, thinking about our EI ratio trait that we found, I think the more nuanced version even than that is that it's not who is the one that's changing, is it human-specific, is it rodent-specific? It's more like, can you actually identify the underlying trait that this change is supporting? Why do you need a change in abundance? Why do you need a change in this molecular pathway? These genes are not just labels, they're in there doing something, right? And so that's really the biology. That's really putting your thinking cap on, or asking Claude and trying to figure out, what is the actual interpretation of these changes I observe? So with that, I'll thank you for your attention. This was a huge effort. I absolutely could not have done it alone. So thanks to my team and all the collaborators who contributed tissue. And yeah, thanks for your attention.
About diversity, we see for species level versus diversity, we see for example, if we take mice, there are so many mouse traits. And they show quite a lot of phenotypic variants, right? Of different code, different even personality behavior. I just wonder, for example, if you like wouldn't be more prudent first to try to scale your approach on variation within the species, like try to research for the community or how different are all breeds from each other on the cellular level before you try to go and make very broad statement about nature. Because like in many cases, you use lab animals which are either genetically impoverished or the environment where they grow up is very impoverished in comparison to wild species.
Yeah, no, it's a great question, and this is, I think, a fundamental question to understand for this sort of work. And so, as scientists, especially if you get trained in one of these, hardcore, molecular biological labs, it gets drilled into you. Keep everything the same, only vary one trait at a time, try to standardize, have everything as replicable as possible. And of course, that's beautiful. That's wonderful, especially for, disease work. I would say you need to do the opposite to some degree for replicability for these sorts of more complex trait analyses. And the rationale for that is when you're trying to keep everything the same, we're just going to test these mice, we're only going to vary this one gene in the lab, you have a mild earthquake or a technician, leaves and another one comes, and the tiniest little variations in the environment of, your species or different labs doing the comparisons will now have an outsized effect. And so actually, it's by introducing more variance that now you raise the noise floor a little bit, and any results you see are actually more robust because now they're more robust to that noise. So you're looking across these different species, and now you're looking at millions of years of evolutionary divergence. All kinds of changes have taken place. But if you can find, nevertheless, despite all that difference, this is the set of genes rising above that noise that is conserved, or this is the set of genes that between these distantly related species has convergently evolved to be expressed in the same pathway. Now you have more confidence. This isn't just a lab artifact. This isn't just, some weird gene, in an inbred mouse kind of skewing the network. This is something that, evolution has run these tests for you over millions of years. These networks have become optimized. And so now I just think that's a much more robust explanation at that point, because you can vary one gene and you can see a difference, but there's so many possible explanations for why that change in the gene is actually affecting the downstream change. You can always say, oh yes, it was because I moved this gene that the change happened. Maybe, but maybe you're driving something else in the system to compensate. And it's that change that's actually affecting the difference. There's so much more that could be happening. So I'm kind of going roundabout, but that would be my recommendation for why looking at these natural species differences, where things, again, evolution has optimized them over time, I think that's a stronger way to link traits to genes. Mm-hmm.
Oh my god. I'm faced with someone who asked me, what's the marker, what's the marker, what's the marker, everything. Is there an example, like a cautionary tale or one example of what was thought to be a marker gene that isn't, that I can relay to make this point?
Yeah, CAR3. Write down CAR3. So there's a cell type, I believe it was discovered in mouse, called an L6 CAR3 neuron. And it has this name, it's replicated across all our taxonomies, except CAR3 is not always a marker of this cell type. So we find the cell type across species. The cell type is conserved. CAR3 is not conserved. So actually that was a terrible name to pick. And I would really strongly encourage you, if you're out there studying cool new species and you're describing new cell types, that's amazing work. Please don't name them after the marker genes, at least unless you do your due diligence to look across a lot of different species and confirm that that's actually a marker across all the species. What you'll find, this is if I was arguing with this hypothetical PI, yes, I get it. You look in this one species, the marker's way up, but it's probably one of these markers that's not quite as strong, but that's probably going to be the one that's conserved. If you think about it, cells, they're doing a lot of the same kinds of processes, right? A transcription factor turns on a suite of genes. Ribosomes do their thing. Neurons migrate. It's, there's a lot of shared molecular machinery in being a neuron or being a glion or whatever. And so if you see a marker that's just going crazy in one cell type, are you really convinced that that's like the fundamental definition of that cell type? Or is it, maybe it matters so little that, it happens to have been going off in this crazy direction and the cell doesn't really care that much because it's not really integrated into the network. This is why you'll see a lot of like link genes, link RNAs. Often they're great markers. What are they doing? Anything? Nobody really knows. Sometimes I'm sure they're doing something, but there is a lot of noise in biology, right? And so a lot of markers I think are, they're a lot noisier than something like a transcription factor or an ion channel that has subtler expression, but it's really integrated fundamentally into the necessary biology for the cell. So, yeah. Yeah, CAR3. Mm-hmm. Go ahead.
Even more like can make a common impression of circuit versus cell. So what you have shown today, makes the argument that actually it's the circuit that matters because the neurons are very well conserved. The actual connection is what matters and what makes a difference between species. But we insist into looking in molecular profiling of specific cells. And the other thing we said is that sometimes to annotate the cell and put it, find different species, you have to change maybe a channel to another one to find a proper functionality, which again comes down to what it's going to do for the circuit. So that's a more philosophical question. Should we more be looking for circuits and how they are preserved and conserved throughout evolution? Or should we keep looking for individual cells, which in the case of neurons, by themselves that don't do anything? They need to be part of a circuit.
So you've just entered a half-century plus long debate about gradualism versus punctuated equilibrium, right? And so, we're looking, I'm describing these changes to you, and it's like, oh, geez, it all looks so subtle. Where are the monsters? Where are the crazy, things that are happening? Oh, well, it must be in the circuits. But, I've looked at the circuits too. And like, once you get over the Dunning-Kruger and once you're really deep in it, it's like, actually, it's still pretty subtle. It's still, just subtle shifts to conserved programs of, like deep molecular machinery. And so I'm more of, what's called a gradualist, where I think it's like there's a lot of recycling of molecular modules, of cell types, like we were talking, Nelson was talking about ICJs earlier. So that, it turns out it's more of like an olfactory granule cell, and the striatum, it needed this certain component. It's like, let's just borrow from something up there that already exists and reuse it. I think there's a lot of reusing and just subtle, shifts that make all the difference, as opposed to, the alternate view, the sort of punctuated equilibrium, like a binary jump happens. That would be like my colleague Eric Jarvis, who, he thinks the vocal learning circuit, there's just like a duplication of a gene that happens. Or, I think Chomsky said this too, and it just magically creates a whole new circuit overnight. I think that's Possibly. I'd love to see the evidence of it. We know now a lot of the genes that I think are supporting things like vocal learning, and it's not an overnight jump. Evolution doesn't know where it's going, right? Like evolution doesn't know that, oh, I'm trying to build the most intelligent ape ever. It's, everything has to be functional right now. Right now, every day is life or death, in the wild kingdom. So, sometimes things go forward, sometimes they go backwards, but anyway, yeah. Mhm.
Yeah, these data sets across species that you may be enriching them in a differently biased way. Because I know like Nelson was talking about how, for example, the mouse data they don't enrich now for the mouse Alex, because you enrich in the highest way, right? But you can also imagine that you're biasing differently in different species. And so when you're then looking at the relative abundances of different cell types, like how do you know what's real?
Mhm. It's very difficult.
Yeah.
It's very difficult. And this is why actually we kept it at the subclass level, which is relatively high. Like if you look at one of our species specific taxonomies, there we're getting into, multiple hundreds of types that are well defined. And we're sure that this, itty bitty cluster is perfectly unique. Once you're comparing across species, it's very difficult. Part of this is it's sampling. Part of this is because, like I said, there's so much transcriptomic drift. The deeper you go, the harder it is to be sure, am I really looking at the same cluster or not? You'll use one seed and get a certain clustering and refine that and be pretty sure. And then if you use a different seed, different set of clusters, it's like, geez, that wasn't it at all. um, and so I will say, one of the trade-offs is the deeper your comparisons go, like if you're going all the way out to marsupial, for instance, if you want to be sure, like you're saying, that these DE differences you're looking at are real, you're going to have to go a little higher up in the tree for your cut of what you're actually comparing. Whereas, if you want to look at a couple different mouse strains and you're really curious about, some specific cluster difference, maybe that's a good example of that would be the right model now for that kind of question. If you really are interested in these super subtle, nuanced differences. Here, for my purpose, I wanted to kind of get the high level, view of things. But, like I said at the beginning, it depends on what your question is. You shouldn't be starting with the model and then asking questions. You should start from your question and then ask, what is the appropriate model with which to answer my question? Right?
But how do you like, how long it takes to optimize the protocol for like tissue isolation, like further transcriptomic work of like these non-model organisms?
Yeah, thankfully we have an incredible person here, Rebecca Hodge, who has really well optimized this protocol for whole brain freezing. And so this works really well for, everything from mouse to human. And it's a cryoprotectant protocol where you're kind of freezing the brain at a slow, controlled rate. If you go too fast, if you just throw it in liquid nitrogen, you're going to get all kinds of, histological artifacts. And so we'd be happy to share that protocol with you. I will say, if you're wanting to do these kinds of comparisons, it would be best to make sure, as much as possible, that things are being collected by the same lab and the exact same protocol, because anything you're shifting, anything that you're changing, that now becomes something you have to worry about in terms of the, just additional sources of noise.
I hope you've been having a good day. Caroline are going to tell you about how some of this work, like for example, in cell typing that we do here, interacts with the broader kind of community and the projects that have been involved in it. And this has been a kind of a long-term initiative of the U.S. Brain Initiative. And the Brain Initiative's focus has been on, was and has been on kind of essentially novel technologies and application of novel technologies in basically brain research. In fact, there were earlier initiatives and people forget about them. There was something in the 1990s called the Decade of the Brain that actually George Bush Sr. was the kind of proponent of that. Barack Obama was the proponent of the Brain Initiative here. But in the Decade of the Brain, actually neuroinformatics kind of got its start there, and some of the people who were neuroscientists first kind of contemplated the ideas of applying informatics big techniques to brain science. In 2014, they wanted to, all these new molecular kind of, technologies had come online, and it became clear that with advances in sort of physiological kind of tools, with molecular tools, et cetera, with epigenetic kind of assays, that one could potentially do a much deeper dive into sort of brain structure. And so like a report was written and people were kind of collected at the NIH and from scientists all over the world. And they wrote up a report. They kind of set the framework for what this brain initiative was going to be. And it was kind of rolled out in 3 stages. Thing was called BICCC, and that stood for Brain Initiative Cell Census Consortium. And that went from just like 2014 to 2017. And the point of that was actually to try out these new technologies, high throughput sequencing, single cell and single nucleus sequencing, to prove that they actually could work on larger scale projects and certain kind of also imaging type things as well. And there was like a pilot, and that the pilot was reasonably successful, and they wrote a paper about that. It was published, I think, in 2017. And that was the little thing you can see up here is the kind of the thing of the paper there. Can you see that? No. Does my cursor move on that? I guess.
Yeah. On the cursor, that's not working for some reason.
Oh, that's okay. It really isn't.
If we go here but now you have a cursor.
Laser pointer.
There you
Wow.
go.
What do you know about that? Yeah. Okay. That kind of what happened is that once that was proven sort of successful, they were ready to launch this kind of in a bigger way. And then the next component of this became what was called the Brain Initiative Cell Census Network. And this was to do large scale whole brain kind of profiling of cell types, primarily in the mouse brain, but with assays and kind of experimental kind of things in certain non-human primates and human to test the waters. And that went from like 2018 to 2022. We finished that. Carol was involved in that. Other people here were involved in that. I'll tell you about some of the accomplishments of that. But in this next phase we're in now is actually that at some point you have to declare victory, right? So victory was declared in the mouse. It's done. Does that mean everything is understood about the mouse brain? Well, no, of course not. But they have to sort of say, okay, we're gonna move on now to the human, right? And so this new phase does continuing mouse work, developmental mouse work, and it also includes sort of macaque and marmoset and human as well. Just some brief things. So a few brief slides here. I don't have a lot of time. I spent a lot of time on that first slide, which gave you the big picture. But this was the BICCN. It was like all these different groups here. You can see over here doing a myriad of technologies from single cell, basically molecular assays, through imaging, through various different kind of tracer type of injection things, and kind of more mesoscale techniques. And there were a lot. It was actually quite a lot of investigators doing a lot of different kinds of things. And so this one of the first accomplishments of this was something called this primary motor cortex atlas. And so what happens in these consortia typically is you start off and you think that you're just going to do this startup project, and the startup project was supposed to be this primary atlas of the primary motor cortex because it had a nice kind of homology of structure between the mouse and the macaque and things like that. So it was this idea that we're going to all get together and we're going to do this. And they called this the mini atlas. And so whenever you start a project, never call anything the mini atlas because you don't have any idea how long it's going to take, you know. And it took quite a while. It took a lot of the time actually of the BICCN to actually do the mini atlas. But it was very coordinated, a kind of working group that basically brought together the molecular side and morphology and electrophysiology through PatchSeq and these various assays to really do a really nice characterization of this structure. Now, it was very challenging to do that in a sense that these groups, you know, there's a lot of kind of difficulty making connections sometimes between, you know, single cell profiling and morphological and tracing and viral tracing. And it's difficult to do this in a coordinated way at scale. And so these groups they sort of split off a little bit, where they went their kind of own ways after this mini atlas. And a lot of work was done profiling the whole brain, but it wasn't really done in the quite same kind of multimodal fashion with everybody involved. We wrote a paper about this, describing this. As you can see here, was in PLOS Biology, just explaining kind of the general structure of the network, the data, some of the things that were done. But in any event, different groups, the molecular people, the imaging people, the you know, kind of the morphological people, they've gone off and kind of explored this mouse brain, the other parts of the mouse brain, kind of independently. But in some sense, the molecular ones was really the one that would probably did, in some sense, the largest scale thing. And here, basically, this group kind of produced a whole mouse brain atlas. Two different groups produced actually a whole mouse brain atlas using these kind of techniques. One was done here with our people here. The other was from the Broad Institute, and I'll tell you a little bit about that. And actually, there was a third one that I should say three, really. It was a spatial transcriptomic atlas of the mouse brain through Xiaowei Zhuang at Harvard. But this led, you know, to a really a very kind of deep characterization of the many cell types of the brain. And, you know, we don't really know, of course, as you probably, you know, you are all becoming sort of experts in cell science. So you probably have some questions about exactly what cell clusters are and whether they're really reliable and where we can draw the line. And that, of course, you know, that's work that's ongoing. And, you know, I calling them cell types is still maybe the putative cell types sometimes is a better word for some of them, you know. So, but in any event, it provides a template for which to kind of compare things and to study things. And this, you know, and basically it's organized typically, as you know, at multiple levels of resolution. You probably heard that ad nauseum today for most of the whole day, but this is, you know, the kind of way the data is organized. And certain parts of the brain organize better hierarchically than others, right? The cortex organizes pretty well. There are some lower brainer structures. It may be a different kind of structure. These were these two big mouse atlases that we sort of brought together from the Broad Institute in here. And a group here, some people in our group did a kind of a comparison, a kind of reconciliation of what, you know, what was kind of produced and reproduced, of course, and corroborated in both, what was perhaps better sampled in one or the other, et cetera, you know. And then this BICCN went on to do these human prototypes, right, and different things like that. And in fact, a group from the Karolinska, Stan Linnerson, actually did a kind of 100 region, very coarse survey of the human brain. But it's a nice paper, Saletti et al., and it's oftentimes referred to. So, you know, these kind of things, the way these kind of pillars, they kind of interact as far as the NIH is concerned. And the three things that we are today are this sort of BICAN, this Brain Initiative Cell Atlas Network, the CONNEX project, which is connectivity across scales, which basically is now the idea of studying connectivity, but also relating it to cell types and understanding essentially what circuit level kind of you know, structure, how it pertains to cell types. And then finally, there's something called armamentarium, which is genetic tools and actually developing these kind of ways to kind of use cell types in their profiling. So there are in this BICAN, there are some very big projects involved, and there's these big centers they call centers that they're sort of profiling different big things. And we won't have time to go through all of them, but you have the slides, you can look at them. But there are several of these, right? There's one here, there's one at the Broad, there's one at University of California, San Francisco, there's one at the Salk Institute, doing all different aspects of it. One of the ones that we have here is this one that kind of Ed Lean and Hong Kui Zheng kind of run. And this was the idea of it's sort of a functionally guided kind of assay where, and it hasn't completely used all the functional side of it yet, but the original intent was to do essentially to get pre-mortem fMRIs which could, identify sort of functional areas in these brains, which would be the same brains used later. They were among terminally ill people, but they would be used later to try to see if there was any association of cell types and function. The project has kind of diverged a little bit now because we haven't really taken so much advantage of the functional side that way just yet. But it's resulted in a really a big standardization. So actually, there's only a couple more minutes left before Carol's going to come up, but I just wanted to sort of say that, so in essence, the mini atlas of the BICAN is this thing, what we call the basal ganglia atlas. And so the group got together and decided that if you're going to tackle the whole brain, what would you do first? And so the idea was to look at basal ganglia, right? And that's because that's something that's evolutionarily ancient, at least 500 million years old. Essentially, the first organisms beyond essentially nematodes have basal ganglia, and they use it it's very important in kind of goal seeking and direction and validating whether something is interesting to go after, right? And so basically there's a big kind of studies centered on that, and we're just writing this up and we're submitting this as kind of another publication package. But so I just want to point out that one of the hopes in this was and Carol will probably tell you a bit more about this, is to really parallel sort of activities and standardization that was done in the human genome towards standardization in cell types, right? And profiling. And here, this is like a kind of a little table here. You can see that it lists a certain kind of standard attribute, which you might like, data formats, semantic standards, etc. And what's done in genome sciences, and then what we're trying to offer here and some of the tools that we've developed here to try to bring those kind of technologies into neuroscience. And it's a very big operation, involves many, many people. There's lots of the processing and the workflow in dealing with the human brain is vastly more complicated than the laboratory mouse. You have, all kinds of issues of more important issues of tissue viability, of kind of permissions, of neuroethics, of scale and size, and all these things come into play. And it was actually a very big thing. And so there's a set of studies that we've kind of done. These are different groups that have done each one of these. And this figure here just shows what species they were done in and what techniques they used and where some of the data is. So this paper, we'll be submitting this paper actually this week. And hopefully if it all goes well, it'll be on bioRxiv, so you'll be able to see it in short order. And this just shows kind of our, everything was very standardized at the point. A lot of kind of standardized pipeline, lots of handoffs, objectives that were kind of identified and managed across groups. I already talked about it was very complexity of the tissue handling. There's a whole kind of group at the University of Texas, a university health sciences in Houston that does a lot of the kind of informatics management of the tissue and sample selection and sequencing. And so one of the things that was derived in this was this really unified kind of ontology of the kind of the basal ganglia. And the idea was that it was done and derived by defining terms we would be interested in from classical neuroanatomy, then looking actually for markers across species in an attempt to identify the same regions molecularly in the different species, and then developing a new ontology which could be used as driving the sampling and the mapping of the data to. And so this gives us an opportunity to do all kinds of sort of imaging, to build these common coordinate frameworks and stuff, and ultimately derive a kind of a new taxonomy, right? And then, so that's what's happened here, is that we have a kind of a cross-species taxonomy in which the cell types are identified in each species, plus which markers pertain to which, and what are the differences in that. And all this data is going to be now available. It's a study of about altogether over 17 million cells across, a single sequenced, across the kind of these aspects of basal ganglia kind of structures. And finally, there's a lot of tools that will be kind of coming out here. And with a lot of partners in the consortium, we've developed a lot of different kind of ways of getting at the data, viewing it, studying it, and providing access. So keep tuned. We hope that this will be coming out pretty soon. And I'll let Carol tell us about what's next.
Okay, Mike's the storyteller. He's telling you about the big picture of the BICAN and the BICAN data, its history. And then I'm the one who, as a kid, took my 400 paper bags and I like to alphabetize them all in a row. This is kind of like my job here in the BICAN. We have a grant together, and a lot of what we do is consortium coordination, engagement, and outreach, right? So this is getting the scientists within the consortium to work together to generate some coordinated outputs, figuring out what everybody is doing, trying to help make it take shape and help make sure people can find it and use it. So my part of the presentation is a little bit more about pointing you to how you find this stuff. And I'm not going to go into a lot of detail about exactly what button you click here and there, but there are a lot of user guides that are available online. But there are different access points for a lot of things. The other thing is that you're going to have a lot of talks today or over this workshop from various people who are also involved in the BICAN, and you're certainly going to see stuff about a lot of the tools that are part of the BICAN. So I'm not going to cover everything because I think you're going to get more detailed presentations about a number of the topics. So this is going to be the overview of what there is and how you find it. So first of all, in the BICAN, we started this in the BICCN, really trying to promote that we want to make the data really accessible. So there's something called the FAIR principles for data sharing. FAIR stands for findable, accessible, interoperable, and reusable. If we want data to have an impact, you want to make sure people can find it, access it, and actually be able to use it. And that takes a lot of different things. FAIR is always kind of an aspirational thing. So FAIR principles, we want to make sure we have identifiers for things. So there's a lot of different places in which we make sure identifiers are issued for things. We want to make sure there's good, rich metadata. So we've done a lot of work on trying to put together metadata standards for the data that's reporting in to the BICAN. And so that's part of the standards as well. And then maintaining that provenance chain for, like, where did this sample come from? Like, how was it processed? And another thing in the BICAN is because we want to make this data broadly useful, we're trying to make it as open as possible and distribute it as quickly as possible. And there's always been a broad tension between you want to make data available, but you want to hold it close to yourself so you can publish on it before anybody else. What we've come to, we started with this in BICCN, and we continue to work with our members and scientists about saying, hey, you know this best, you've generated your data, you're going to work together, you have a ton of data, you're going to get your conclusions, you're going to get your papers out. It's not going to hurt you to share it. And so we have policies for sharing data rapidly. Now, a lot of the rapid data sharing ends up being very base level data, the level, what we call level 1 data. So kind of the raw FASTQ and the BAM files that have been aligned to the genome, and then some of the imaging experiments. And people who know what they're doing can go in and use it. But taking the time to turn that into more easily accessible information is a lot of work. And so that is what kind of gets delayed. And it's adding that knowledge layer and that expertise to it is what really ends up giving us where we're going to learn about what cell types are, and that's the stuff that goes into those publications, right? So I encourage everybody as you do your science, if you're doing like big projects like this, there's so much data to release. You should be able to release it openly, and you should be able to release it early. Your individual little experiment you did here and there to do your hypothesis testing is a different story as you're trying to get to your publications, but we're trying to make sure everybody can use this data. And the other thing we've been doing is trying to make sure that we can share under the least restrictive licenses possible. And so we've done a lot of work in the licensing space to look at this. If things are not human sequence level data or other things that might fall into some category where you're worried about de-identification, we've been in the consortium for the raw level data. We've been trying to make sure it stays as a Creative Commons Attribution International License, which is basically CC BY. This means you can use it, you can reuse it, mix it up, make derivatives, but you have to give attribution to its source because that's our scientific custom to make sure that people can reuse data, right? But when you get into some of the human data, it gets a little trickier as you're trying to make sure that you are reducing the risk of re-identification of donors, et cetera. So we actually have a bespoke license for that, our BICAN by NR, which includes a non-re-identification clause as well. And then higher level products may have other considerations. You always have to check the licenses that are on any other higher order knowledge products because you don't know what else has been remixed with it to make those products. And so licensing is a little ad hoc there. Okay, so in the BICAN space, for the types of products we have, when I'm trying to chase after everything and say, what are we generating and how do we make it available to everyone? We have probably 5 broad categories of these things. So there's standards and documentation, and we have a lot. I'll show you in a moment. So the standards and documentation, we think a lot of these standards are useful to people in terms of being a good, not only documentation of the work we've done, but examples to other people of what kind of standards you could use. There's data sets, of course, this is the big product. And so we have a way to organize it, our data catalog, for that librarian sense, like what's all the data? This is one that's we're continuing to develop into. There's a lot of data. We're continuing to try to make sure everything gets represented there. Data gets released to data repositories, and then we have some concepts of data releases. There's atlases, digital reference atlases. This might be cell types like Nelson was making, these cell type taxonomies, where you're clustering the data and figuring out what the cell types are. And there's anatomic atlases. So how are we making these common coordinate frameworks for the brain and that are being annotated with anatomic structure names. There's publications that are coming out, and these are an important part of really explaining and distilling what the BICAN is finding. And then there's other tools and infrastructure that make this stuff possible, and in some cases are also access points and reusable. I have not listed every single thing under each of these categories. There is a long list. Mike showed one slide that showed a lot of resources, but I'm just going to give you a brief spin through some of these. Okay, so in thinking about our tools and infrastructure that support the BICAN space, there's the Neuroanatomy Anchored Information Management Platform that Mike mentioned, NIP. This really represented a big change in the way BICAN was organized to the way BICCN was organized. In BICCN, every lab was alone generating its own data, and then it would come time to, okay, can you organize your data and upload it to a repository? And sometimes a lot of metadata got lost in that process, and sometimes things couldn't be synced up. So in the consortium for BICAN, a major part of it that was built in was this NIMP platform now that is organizing not only the metadata, it organizes a specimen request, it organizes the metadata about them, metadata down to when you generated a library to be sequenced. It organizes some of that, the fact that the sequencing was done. As a central place, it captures the entire provenance for the single cell sequencing, not for the spatial transcriptomics necessarily, but it really means that everything is there. You don't have to back enter a lot of stuff. There's some curation and cleanup to do. But it's really been a game changer this time around to have everything going into a central platform from all these different labs working on different species with different omics profiling techniques. But it does mean that at the end, you can actually capture all that information. A big thing that the NIMF system does, that's important to think about when you're looking across everything, is that it's generating identifiers for each of the entities. So if you have a donor, there's a NHAsh ID for the donor. If you have a piece of tissue that came from that donor, that has an identifier. If you have a slab, you have a section made, you have a dissociated cell sample, and then an enriched cell sample, and then a barcoded cell sample after your library generation technique. And then you've got a library and a library aliquot. All of that is tracked. Each piece of that has an identifier, and you can track it down a provenance chain. And the NIM system is going to make their a version of their tool that manages all this metadata publicly available very soon. And so you'll be able to go in and view the metadata and generate these little plots here that show from a donor here at the left, all the way down to the different samples that were generated from it. Usually the tree is much bigger. This is just a small example here showing you all the upstream and downstream components, it looks like, of this one barcoded cell sample here. So, and in the BICAN, there were also a lot of pipelines generated so that data could be processed in a standardized way. These are pipelines that take the FASTQ files, align them to a certain number of genome references, and then generate the BAM files, as well as QC metrics and other artifacts. And so a lot of these have been generated, and there's a lot of great documentation about them. People can reuse these pipelines if they have data and want them to be generated in the same way. Okay, so for BICAN data releases, to get to the data sets, as I said, BICAN's committed to open neuroscience, rapid sharing, and yet we've been a little slow. We had our first rapid release in 2024, and we're just getting ready to do our next one because we've spent a lot of time trying to do the automation in the back end. The BICAN is a very distributed ecosystem. So there's that NIMP platform, and then you have the individual labs that generated data who need to check in and make sure things are right, and sometimes back enter things and fix their data. You have the data repositories, and then you have our catalog system for searching the data, and getting automation for the data flow to go between those sometimes takes a little time. But the description of the process we have right now for the omics data is that you have a continuous release process where this is relatively automated. Data is generated, gets logged, the scientists review it, they say it's good, at least at a very first pass level. It has to really say data is really good, you need to do a lot more analysis. So there's like an initial set of QC where it's like, okay, this looks like it's useful data, you got enough reads, you don't, whatever else, you're looking at a bunch of different metrics there. And so these data then flow their way through to a public directory. Then we have a concept of a rapid release where we actually package the data a little bit more. And so this is after that basic QC, and we are making sure we've got citations for everything. And we wrap it all up, and then it will be searchable in the data catalog. Eventually, we expect it to be iteratively refined and through analysis and adding on those knowledge layers. And then we see some of it going out in the ABC Atlas right now, and some of this may go out in other knowledge-aware tools as well. And then the centralized data catalog allows you to search for and find the data that have been released, both, and I'll show you those examples in a moment. Okay. So the data catalog is our way of organizing this. This is part of our knowledge.brain-map.org, which hosts a lot of data for the Allen Institute as well. It hosts data for BICCN, BICAN, and Microns, and the Seattle Alzheimer's Disease Consortium and probably some other ones I'm not remembering off the top of my head. But what we do have in there is we have a BICAN program page that you can start on. I think it's been updated since this particular photo. And from there, you can see what projects are in it. And then if you go to the individual projects, you can search within them for the metadata for this particular specimens that are represented by the data in those projects. In the BICAN, these also link out to the different data repositories that host the raw data. And by the design of NIH, by how they wanted this brain initiative work to be hosted, we actually have our data archives are separated by data modality. So we've got NEMO for the transcriptomics and epigenomics data. The BILL brain image library is for imaging data. So we've got some of the spatial transcriptomics at BILL, some of it's at NEMO. The difference would be if some of our spatial transcriptomics data is sequencing based and some of it is imaging based. And so the imaging based data goes to BILL. And then we have the DANDY archive, which is a distributed archives for neurophysiology data integration. And this hosts for this consortium, our PatchSeq data. It hosts the electrophysiology component of our PatchSeq data. PatchSeq is actually split across 3 archives. PatchSeq is one where you take a cell, you do your whole cell patch experiment, you record the ePhys, that goes to Dandy, you get a morphology on some of it, that goes to the BILL data archive, and you have transcriptomics for it as well, and that goes to the NEMO archive. And so we're continuing to work on better ways to package that data so that it is more easily accessible altogether, but at least it is unified where you would be able to find all 3 collections of data at the different archives by looking in the data catalog. And the BOSDB archive is one that was also used during the BICCN, but for BICAN, we don't actually have much data going into BOSDB. Okay, so how do you go find this stuff if you want the data? So again, there's different levels of the data products. Some of it is the raw data. So if you know how to use that, you'll find it there. Some of it will be analyzed data. A lot of different groups are starting to put their data into different viewers. And this is where we're still working through indexing all the different host sites and how they're hosting that data. But for now, what you can do is you can start, one of our starting points is you can start from brain-map.org. There's a consortia page, and you can see there's a BICAN here and a BICCN there. You can click on some of these consortia to get to a page about that. And this particular page will take you to information right now about the rapid release inventory. But we'll probably, and it has links to other related resources right here. But we'll probably update this as we go along and add links out to other resources in the future. So as I had said, this page shows you the rapid release inventory and takes you to links where you can find out about this. Our first rapid release was in September of 2024. This included data from just 6 labs, 255 donors, 1,461 specimens, and 20 data collections. That will be a lot more in the release going out. We have more donors, more specimens, more labs, more collections. But right now in the catalog, we still have the September 2024 set. But if you were to go, you can also access the rapid releases directly from the NEMO site. And right now they have a landing page. I guess it's really small, sorry. That is everything in the rapid release this time, which means all the single cell omics data is listed here. And so what you've got is each of the NEMO data sets has a collection ID up at the top. We will index these collection IDs from the data catalog. There's one big one for the whole rapid release. This is for the 2026 rapid release, and they put this package together this year. And then there are individual, sorry, down here at the bottom, there are individual child collections. So at the bottom, each one of these will have, if you click on them, you'll go to a different page and they'll each have titles and they'll have authors and grant acknowledgements. Each of those are divided up by type of experiment. So whether it was a tax-seq, multi-ohm, whether it was what species it was, marmoset, macaque, human. And we also divide up the collections that might have controlled access data versus open access data. One thing that we have done in the BICAN is a large majority of our human donors were consented for open access data. So a lot of them, you can get the data without having to go through a controlled access system. But if it is a controlled access consented sample, then you will have to go through a process through the NIMH data archive to obtain access to that. And that'll be through the email site. Okay, so another access point you can come to anytime you want to find out more is the BICAN portal at brain-bican.org. And here I'm showing a rapid release page, which is detailing that last release. And we'll update this with the next release that is about to go up. And so here you can find links to all the different things. There's a lot of information on this page. It'll talk about who the contributors were, how to get access to things. There's links to the BICAN program page, links to the catalog, links to the release notes. There's a tutorial on how to download a file manifest for all female chimpanzees from this particular grant, and then how you would use that to download all the data files from NEMO directly. And then there's some other user documentation on the release. Okay, another access point. There's so many different ways to come into this, which is wonderful, but can be, you know, in this presentation, I'm like, well, it's a lot of different ways you can go find the data. If you were to start from the data catalog, which again is hosted in that knowledge.brain-map.org, URL's down here, you can come into there and you can search. If you were to click on programs, you could get to the Brain Initiative Cell Atlas Network right there, and it would open up a program page that would show you all the different projects that are here. And you could click on any one of these projects to go find more information about those projects. It is always a dynamic process of updating this. Sometimes we have projects that are being registered, but the data links weren't ready yet, or the data wasn't ready yet there. So it's always a process of putting these in and then coming back and following up with, okay, now all the data links are ready. Now we can add those. So if you see the entries there and there's no links to the data, they weren't ready, but they will get populated in a future update. Okay. So on some of the different project pages will also be extra links that will take you to go to different resources, like how to use the file manifest to get the data at NEMO, or how to get to the BICAN specimen portal, the NIMP specimen portal. Okay, and this is an example of the page where you can go that Sven put together to figure out how to use the information you can get from the project page and the list of specimens to go download your data from NEMO. Okay, now this was data sets. Now I'm going to switch gears to digital brain atlases just briefly. There's actually a really nice access point now on our website at brain-map.org. The URL is down here at the bottom. If you go to consortia, the HMBA is our Human and Mammalian Brain Atlas Consortium, and that is part of the BICAN Consortium. And they have a nice page up here that details a lot of the tools that are being generated and a lot of the knowledge that's being generated out of this effort that as part of the BICAN. So there's that cross-species taxonomy information. If you go here, you can click through and you'll get to a page that tells you about the consensus taxonomy and how to get all the files. There's also a link out to the anatomy resources that we have right now, such as that harmonized ontology of the mammalian brain anatomy. we call this HAMBA. And HAMBA is, you know, if you've looked at different brain atlases, every person who makes an atlas has a different terminology. Every person who makes an atlas will draw them in a different place, and every species has a different set of brain regions. Even if, you know, generally you say there's a thalamus, there's a cortex, there's a hippocampus. What was done in this one, the novel thing that has been done here is that it's been harmonized across, it's meant to be useful for all mammalian species, but right now we're fairly, it's set up for BICAN for usage in human, marmoset, macaque, and mouse. So if you go to this page, you can find link outs to the HAMBA ontology, which was recently also deposited onto our brain-BICAN GitHub repository. So there's a new repository in there for the HAMBA ontology. There are also, I think, link outs to some of the atlases as well in 3D. Okay, standards and documentation is an ongoing thing. We have, during the course of the BICAN, done a lot of work on putting together a standards governance framework. And so, and metadata standards in particular go through this process where we've been gathering expert provided lists of metadata that they want. There's been a process of doing this integration and mapping them to terms, and then making sure that we can document all the fields that we want to map, and then they are put into the NIMP platform for organizing that metadata per sample. And we have a review process where it goes through a couple different committees for review. But if you look over here on the right, this is an index of many different standards that we've looked at or considered or reviewed. Some of them have been formally reviewed and some of them have not. So we're still compiling and curating and running everything through an approval process in that. But you can go to our GitHub and see our working documents at github.com, brain-BICAN metadata schemas listed down here at the bottom. And we're currently continuing to further integrate a lot of our metadata standards here. Okay, and then if you want to find the latest science from BICAN for the publications, there's a couple different places. At brain-bican.org, we have a science page that has both the BICAN and the BICCN publication packages. So each one of these listings here is a set of many papers that were all coordinated at the same time, detailing a lot of the things that Mike covered back here. So I think down at the bottom would be the motor cortex. Then we have the human and non-human primate brain, and then we've got the whole mouse brain atlas. And then the most recent one we have out of BICAN is the developing mouse brain atlas. This is really based on the BICCN work, but a number of groups coordinated analysis and publication of work highlighting what they're finding from the data in the developing brain. So I encourage you to go. You can click through and look at that set of papers in Nature. But if you really want to find the newest stuff that's not published yet, we have a channel on bioRxiv on the preprint server. So you can go to the BICCN BICAN channel. It's got 72 total articles, and then the newest ones are BICAN. It has a lot of BICCN articles as well. But all of the basal ganglia work that we've been working on recently, each paper as it's getting submitted is feeding into here. So this is stuff that is hot off the presses, right? So these are things that started being submitted in about December, and new manuscripts are still trickling in, but a large tranche of them are in here in the preprint server now too. Okay, with that, it's a thank you to contributors to our first BICAN rapid release, which were all these grant awards here. And big thank you to NIH and NIMH for funding this work. We're really excited to be able to share this with you. If you have questions, please let me know.
My name is Scott Daniel. I'm Same team as Chris here at the Institute. Yeah, I'm going to talk to you about the MapMyCells tool. Maybe this next slide is redundant given what Rachel just said, but I just want to give a quick intro. What is MapMyCells? MapMyCells is a tool we built in the Brain Knowledge Platform ecosystem to take cell by gene data or anything that looks like cell by gene data, by which I mean anything that is just a row, a matrix where rows are cells and columns are genes, and then annotate the cells in that matrix to the various cell type taxonomies supported by the Brain Knowledge Platform. Right now, those are the whole mouse brain taxonomy from Yao et al. 2023, the whole human brain taxonomy from Saletti et al. 2023, the cross-species basal ganglia taxonomy that got published late last year, this year, depending on what you believe about preprints, and then the medial temporal gyrus taxonomy from the Seattle Alzheimer's disease atlas group. So those are the taxonomies supported by MapMyCells, specifically by the web tool. MapMyCells is two things. In a more granular sense, it is a web tool, which I will show you in just a moment, where you can go and upload your data and get it annotated to one of these 4 taxonomies. It's also the Python library that runs the algorithm under the hood. It's an open source library. It's designed to be run by users such as yourselves. You can clone that code and you can run it. And I'm just drawing attention to this because there's software in there that allows you to run MapMyCells on any taxonomy that you have. If you have your own taxonomy and you want to annotate new data to it, there are functions and example notebooks in that GitHub repository that will show you how to prepare and do that process. Please talk to me about it later. I love talking about nothing more than I love talking about this. So I'd love to show you how to do that. How do you use it? It's really simple. I'll do a demo right after this slide, but really MapMyCells, and again, I'm kind of more talking about the web tool here. It takes either an H5AD file, which is a format supported by Python and data library. It's getting popular. Hopefully you've heard of it, but it also takes CSV files. You can just give it a CSV file. Again, rows are cells, columns are genes. You can upload it to the web tool and make a few selections and start mapping your cells onto the taxonomy. I keep highlighting this point, rows are cells, columns are genes. That's what we would like. However, if for some reason your data comes the other way, if rows are genes and columns are cells, it will do its best to transpose it, and it'll probably do it right. If you put too much data in that form, something on the back end might run out of memory and you'll get a crash. But generally speaking, you can just give us a CSV file, we will map it. And that's it. The only other requirement is that the file you upload to the web tool has to be 2 gigs in size or less, and that is a requirement imposed by your browser. You upload the file, you click some buttons, and you're good to go. It's even capable of taking data from one species, for example, taking human data and mapping it onto a taxonomy from another. You can map a human data onto your mouse data, onto the mouse taxonomy if you want. It just uses lookup tables downloaded from NCBI to find orthologous genes between the 2 species involved. So really, we are remarkably agnostic about what kind of data you bring to us to map with MapMyCells, which is what I'm going to show you now. I'm going to be a lunatic and do a live demo because that always just works. What I did before coming here was I went to cell by gene and I just looked for some data that might be interesting to map. And I found this data set, molecular and cellular evolution of primate dorsolateral prefrontal cortex. And then looks like they've got some human data, they've got some chimp data, they've got various flavors of macaque. I decided to download the rhesus macaque data, again, because I'm a lunatic and I want to show off what we can do. And so now I will take this rhesus macaque data and I will map it to a mouse taxonomy because you can. No, this is not from us. Yeah, sorry, maybe I shouldn't have been so glib about that. Yeah, this URL is really small. cellxgene.cziscience.com, cell by gene. Lots and lots of data from all over the place. Yeah, so MapMyCells, the web tool. You go to brain-map.org, cell types and taxonomies, MapMyCells, and this gets you to the landing page for the MapMyCells ecosystem. Here we have links to a bunch of examples and documentation, more than just the app. So this is where you would go to help learn about what MapMyCells is actually doing. I'm going to just scroll down here and go to the actual app itself. And so this is the page that you see. That's too big, Scott. So first, you have to upload some data. So I'm just going to click here. And here, like I said, I already downloaded it before I came. When you get it off of Cell by Gene, it has a big uninterpretable name that has a unique ID buried in it. I just renamed it to dorsolateral prefrontal cortex macaque .h5id. So I'm just going to click open, and it's going to start uploading the data. And this is just now we are just throttled by the realities of the modern internet. Come on. It will take a CSV as well. Yeah. 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 Mhm. So come on.
Is each cell mapped individually or context also matters? I wonder like if I have data set that is still unQC'd, is it worth mapping it before QC?
Every cell is treated individually.
Yeah.
Every cell will be treated individually. Once I get this kicked off, I'll do a brief little talk about what it's doing under the hood. But there's also a link in my slides to a different webinar where I go into more belabored detail about what it's doing. So you can learn about what the algorithm is doing under the hood. So yeah, our file has uploaded just under the 2 GB limit. So now I go over here and say, okay, what do I want to map it to? I promised you I would map to a mouse taxonomy. So here are the taxonomies we can choose from: whole human, human MTG, consensus basal ganglia, whole mouse. There's some algorithms to choose from. I'm going to just choose the default one, the hierarchical mapping algorithm, and then I click the big friendly start button. When I did this at home, it took about 7 minutes. So I'm going to go talk to you now, and we'll see if it's actually done by the time I'm done talking. Otherwise, I did download the results earlier. So what's going on now? Okay, this QR code will take you to a webinar on YouTube where we described in detail what the algorithm is doing. I'm just going to give you a brief sketch here because I was told to limit my time to 15 minutes. But yeah, feel free to watch that if you're curious. What MapMyCells is doing, so the first step it's going to do, because we gave it macaque data, is it's going to look at the genes in our macaque data. It's going to look at the genes in our reference taxonomy, realize, oh, that's mouse. It's going to find ortholog macaque gene or ortholog mouse genes for the macaque genes in the macaque data, and just rename the genes in the data set to be the mouse ortholog so that it can actually compare the two data sets. After that, MapMyCells treats every taxonomy as a hierarchy. Yes. Not anymore. Not anymore. Used to be I would get up here and jump up and down about how you have to give me an ensemble ID or I cannot map your data. The same tool I just described that does the orthologous mapping, it will also, if you give it gene symbols, it will look at those gene symbols, it will compare the NCBI tables and say, oh, these gene symbols are mouse, or these gene symbols are human, or these gene symbols, that's either mouse or human. And then it will translate them into the represent ensemble IDs. If they're duplicate gene symbols, those will get mapped to a nonsense value because it doesn't know how to break the degeneracy. And then it will be subsequently ignored. It will ignore duplicate gene symbols in your data set. The best thing to do is give us ensemble IDs, but you don't have to. Yeah, thank you. Yeah. User feedback. So our taxonomies are all arranged hierarchically. The whole mouse taxonomy to which we're mapping now, there are classes, and there are subclasses which are all children of those classes, and clusters which are children of those subclasses. And so what MapMyCells does is it has a structured table of marker genes. For each level in that taxonomy, it has a set of marker genes that are good at distinguishing between the children at that level. So it grabs the marker genes that are at the root level of the tree, uses those to determine what class the cell belongs to. And then for each class, it grabs the marker genes associated with that class, which are the marker genes that are best tuned to selecting between the subclasses that are children of that class, and then it selects the subclass. And then at each subclass, it selects the marker genes associated with that subclass and uses those to assign the cluster. So it just walks down the tree, assigning the cell at each level of the tree. It also does one other thing. It makes each of those assignments 100 times. So at the root level, it will map each cell 100 times, each time using a different random subset of the marker genes. The idea being it keeps track of how many times the cell is mapped to each class, and then picks the class that is mapped to the most times. This is a way of measuring the robustness of the assignment. The idea being you don't want to be too sensitive to just like a handful of genes. So you randomly subset your marker genes, you do the mapping, you randomly subset again, you do the mapping, and you see if it is consistent. If it is, our thinking is if the mapping is very consistent, if all of the iterations map to the same class, you can trust that mapping. If only 50% plus 1 map to the class, it's kind of suspect. And then you can have really pathological cases where the majority winning class was only mapped to 25% of the time, and then you have a bunch of different classes that were mapped to 3% of the time, and then, buyer beware. So it's just a way for us to kind of measure the quality of the mappings coming out of MapMyCells. And so it just walks down the chain and, yeah, and just keeps going until every cell is assigned to every level of the taxonomy. How well does this work? We have done some test train holdouts on the various taxonomies that we support. These figures are taken from the brand new MapMyCells preprint, which is on the bioRxiv as of, I guess, yeah, was it March? I guess it was March. So yeah, again, if you want more detail, you can go to that DOI. These figures just show 3 taxonomies, the Yao et al. Whole mouse, the Saletti et al. Whole human, and then just for kicks, the Langley et al. Whole mouse, which is not actually supported by the web app, but is a whole mouse brain taxonomy from the Broad Institute that we ingested using the open source Python library that I mentioned to you earlier. And so we just divided each data set into a test set and a training set. We use the training set to derive marker genes, and then we map the test set back to the taxonomy. And this is the measure, this is the performance that we got. And you can see that at the highest levels of the taxonomy, it does very well, almost 100% accurate. Middling ranges of the taxonomy, 90% accurate. And then at the very finest level of the taxonomy, of what we call the clusters, 80%, 70%. But these clusters are more computational artifacts. It's not clear to us that they have biological meaning. This test is great. It belies some things in that the test data was taken from the data that was used to drive the taxonomy, which means all 32,000 genes from Yao et al. Were in the test set for Yao et al. But not everyone has a 32,000 gene panel, or maybe you have a different 32,000 gene panel. So we also did a test where we took our test set and we randomly downsampled it to have fewer genes than in the training set. And so in these plots, you see, again, the F1 performance, this time at a single level in each taxonomy. And the horizontal axis is the number of marker genes in the data set. We're trying to simulate here, for example, a spatial transcriptomics scientist who can only come to us with 500 genes, maybe. And what we see is, so the blue dots are just a random selection of marker genes, literally just drew genes out of a hat. And actually, the performance is quite bad in that case. But the orange pluses and the green crosses are assuming an intelligent scientist who picked their gene panel to include genes that are good at discriminating between cell types. And if you just make that simple that simple trick of being intelligent and choosing useful genes in your data set, then with as few as 500 genes, you can get almost as good performance out of MapMyCells as you can with the full 32,000 gene panel from the single cell transcriptomics data. Final figure I just want to show you from our paper. I talked to you about the way MapMyCells does the mapping and how each mapping we do it 100 times, randomly subsetting the marker genes. That allows us to produce quality metrics. So these distributions show 2 quality metrics from our mappings. The left column is what we call the bootstrapping probability. That is the fraction of those random iterations that picked the same cell type. And then the middle column is the correlation coefficient between the cell and the chosen cell type. And the 2 distributions are for cells that were correctly labeled and in orange, and incorrectly labeled in blue. And the takeaway is just that bootstrapping probability, that fraction of cells that were that fraction of iterations that chose the same mapping, is a very good measure of quality of mapping because the distributions are very different. You get much lower values for incorrect cells than correct cells, whereas correlation, it's not quite that clear. So just wanted to draw attention that there is a way within MapMyCells to tell whether or not you can trust a mapping, and I would argue it's that bootstrapping coefficient that you get. Okay, I've bloviated long enough. Let's go check on our demo. Hey, what do you know? Completed. We've mapped macaque data onto the mouse taxonomy. So then what I would do is click on the download results button, but I've already done that. So I'm just going to show you what I would get because I don't want to, there are reasons I don't want to do it again. I'm not a biologist, so I don't know. I decided to do something hard because I thought it would be fun. I'm not going to say. I don't know. It depends on what you want to get out of it. Someone has raised their hand.
Have you tried something good? Number of people. Something that you like this method will examples.
Yeah, simulating different noise profiles is a thing that we know that we should do. We haven't come up with a good way to do it. Or like a way that we all agree is the right way to do it. So I don't have a glib answer to your question, unfortunately. I do know Jack Water. Is it Jack Waters or Jack Water? Is there an S at the end of his name? I know, right? Yeah. Jack Waters here at the Institute has been doing a lot of work on that with regard to our spatial transcriptomics data, like trying to take single cell data with a ground truth annotation and munge the noise profile so that it looks like spatial transcriptomics data and see how good the mapping is. I'm sure if you reached out to him, he would have intelligent things to say about that question. I can put you in touch if you would like. Yeah. So if you if you click the download button, what you end up getting is so I've already downloaded the results here. Yeah. You actually get where is it? You get a zip file that contains all the rest of this data. So you have some file with some metadata in it. You have validation log.txt, which just is any messages that were printed to standard out over the course of the run. So just kind of keeps track of what's going on. Okay. Sure. Yeah. Where even am I? Yep. So yeah, I guess. Yeah, so like that's a log. That's what that was what was just getting printed to the screen while the thing ran. But I'm gonna draw your attention to this. There'll be a file. It'll be the name of your file, the taxonomy you map to, the algorithm you chose, and then a timestamp to make it unique, .csv. And that's the results of all of your mapping. Yeah, I'll open it with Excel. Open.
They're all going deterministic or there's some.
It's deterministic in that in so much that there is a randomness, the random numbers generator is seated at the front. So if you run it twice, it'll do the same thing. Yeah. And so what you end up with is a CSV file. The first few lines are just some metadata about the run. What was the file? What does the taxonomy look like? What algorithm, and more importantly, what version of MapMyCells where you run. And then you get a table where every row is a cell. And then you have assignments at every level of the taxonomy. You have the class assignment. You have that bootstrapping probability telling you the quality of the assignment. You have subclass assignment, bootstrapping probability there, super type, and cluster assignment. And so now you just have a giant CSV file that you can read in with pandas and or Excel or however you'd like to read CSV files, and it maps your cells to the taxonomy. Let me just show you the results of our ridiculous experiment mapping macaque data to a mouse taxonomy. So this is just a notebook I put up. It's in Rachel's Spocked repository that just takes the result of that mapping. Yeah, so the data we downloaded really quickly came with its own subtype, subclass, and class annotations. These are not guaranteed to have anything to do with our classes and subclasses, but whoever published this data set annotated it with their own cell types, and that's important because it means that I can produce a confusion matrix. And so I'm just going to scroll down, get to the punchline. Code, there we go. This is the horizontal axis is the mouse class found by MapMyCells, and the rows are the vertical axis. These are I guess they call them subclasses in the data, the macaque subclasses. And you can see there's some diagonal, it's somewhat diagonal. There's a pretty good correspondence between the types. And then if I do a filter on that bootstrapping probability, so I'm going to filter it so that I only accept cells with a bootstrapping probability greater than 0.5, I lose about 11,000 cells, and my confusion matrix gets a lot cleaner. And you start to see, immune cells go to immune cells, vascular cells go to vascular, it did the right thing, even mapping across species. I can go down one more level in the taxonomy to the MapMyCells subclass and the macaque subtype. I'm not going to label it. There are too many of them. The axes would be terrible. But again, and this is putting that filter on. Out of 130 cells, 112 pass my quality cut, and this is what the confusion matrix looks like. Yes. Yeah. Sorry. I should have probably thought of that. That's a song and dance I prepared. Questions? You just want to play around with it. Yeah. No, you can't. Sorry. Yeah, it has to. We understand there are a lot of people who want to map with bulk RNA sequencing data. We need it needs individual cells in the data set. We don't have anything attached to it to deconvolve the bulk data and attempt to predict anything from the individual cells in the bulk tissue. So. Yeah. Yeah. I mean, so if you can split your data up into 2 GB chunks, you can map them each individually, and then you will get each one will have its own CSV table. And I mean, the CSV tables are just, every row is a cell in the same order as in your file, so you could just stack them. Yeah. Right. Yeah, you can also clone the code, the Python code, the open source library, and run it for yourself locally. And then the only limitation is there is no limitation. There's always a risk that you will overrun the memory on your computer, but there are parameters you can set to tell it to only process 10,000 cells at a time, so it doesn't overwhelm the memory. So yeah.
Probabilities you record stringent.
Yeah, I have spent an inordinate amount of time trying to get that probability to be an actual probability so that 50% of the cells with 0.5 are wrong and 50% are right, and it's not. It's close, but it's not. So it really, you do have to do some qualitative. It's a very qualitative choice that you're going to have to make depending on how many cells you're willing to sacrifice and how much quality you're willing to sacrifice. I have found, so if you do a mapping job and you treat the probability as though it were a strict probability. So like if a cell has 0.6, then there's a 60% chance of it being right and a 40% chance of it being wrong. If you use those values to estimate how many false positives are in your data set, it underestimates by a factor of 2. And it's a fairly consistent factor of 2, and I can't explain that. But that might be something you could do. So it's like, do that kind of statistics and see how many false positives would I be accepting if I did this? Am I comfortable with that? Yeah. Yes. Yeah. If you just Google cell X gene, well, fine. It's just not completes. Can I make that any bigger? Nope. cellxgene.cziscience.com. You see that? Yeah. It's kind of a big repository for cell by gene data.
And some functions on their tools that our tools cannot do. So if like, if you're looking at data in the ABC Atlas, if you want to like really differential expression, I don't know how well it works. It has, but they do have a tool that does that. So there are two things to use complementary. Yeah, you can find our data on there as well.
The low confidence score, can it indicate the doublet?
Yeah, it could. I'm trying to think if I've ever, I've never actually done that test where I've run it through on pre-QC data, or like data that had been QC'd, but run all the data that failed QC through and see if the low scores correspond to specific QC failures. I haven't actually done that test, but it would just indicate, yeah.
Second best category. Data.
So yeah, if let's see what happens. I think if I open this, it's going to go, it's too big, but let me try opening something in my browser. So there is one other, you're also, when you download the results, you're also going to get this .json file, and that is a more detailed set of results from MapMyCells. See what happens. I think my browser's not going to like it. It's too big. No, it is. Hey. And what you would see there is this list for every cell. And so for every cell at every level in the taxonomy, it gives an assignment, the probability, but then it also gives runners up. It gives like the next 5. If there are 5 cell types that it thought about but didn't choose, it will give you those as well. But that involves working with this JSON output. In the GitHub repository, there's documentation there, and there are example notebooks that show you how to do that. And I'm happy to help you with that if you want to play with that. But yeah, it does this JSON file has a more detailed output than the CSV file does. Yeah. So the way we map to the whole mouse brain, the whole mouse brain taxonomy has 4 levels: class, subclass, supertype, and cluster. But for historical reasons, we found it's better to just go map to the class, the subclass, and then directly to the cluster, and then infer the supertype from the cluster. And so that's a flag I put in there, just indicate here. So it's true for all the levels except supertype. It is not, because the supertype, we didn't assign the supertype. We assigned the cluster and inferred the supertype from that. Yeah.